Hi, my name is Lauren Klein, and in this video, I'm going to discuss my responses to the case studies for Unit 2. To start off, case study number 2 covers the regulation of glucose uptake by the cell. In normal tissue, glucose uptake is regulated by many tissues in different ways in the fed and fasted states. I'm going to begin by talking about the uptake of glucose in normal tissues of the liver, skeletal muscles, brain, and adipose. Starting off with liver metabolism in the fed state. In the fed state, glucose will be the primary fuel oxidized in the liver. The uptake of glucose is an insulin-independent manner by GLUT2. GLUT2 will always be present in the cell membrane, no matter if insulin is present or not. In the liver, glucose is used in several different ways. First, in the fed state, glucose is taken up and used for glycogen synthesis. Glucose is then oxidized through glycolysis. Glucose also enters into the pentose phosphate pathway, as well as the tricarboxylic acid cycle, which is known as the TCA cycle, after it's oxidized through glycolysis. Glucose in the liver will also activate cholesterol synthesis. This also activates the urea cycle as well by disposing the nitrogen from transaminated amino acids. Glucose also activates fatty acid synthesis, which allows us to package fatty acids into triglycerols. The triglycerols are then packaged into VLDL particles, which enters into circulation. Next, I'm going to discuss the skeletal muscle. So, the skeletal muscle metabolism in the fed state uptakes glucose also in an insulin-dependent manner, meaning it only uptakes glucose when insulin is present. The primary glucose transporter for the skeletal muscle is GLUT4. There are several pathways in the skeletal muscle. The skeletal muscle will oxidize glucose through glycolysis. Glycogen synthesis stores glucose as glycogen in the muscles. In the skeletal muscle, we'll have amino acid uptake from protein synthesis. The TCA cycle is active and is oxidizing acetyl-CoA in an aerobic manner. In brain metabolism, in the fed state, glucose is taken up in an insulin-independent manner by the primary glucose transporter GLUT1 and GLUT3. In the brain, glycolysis takes place. Other than starvation, the brain will primarily oxidize glucose. In the fasted state, in the brain, glucose is taken up in an insulin-independent manner and oxidized through glycolysis, once again by GLUT1 and GLUT3. There are two pathways providing the fuel in the brain. These pathways are hepatic glycogenolysis and gluconeogenesis. Adipose metabolism in the fed state will uptake glucose in an insulin-dependent manner. This will happen through GLUT4. In adipose metabolism, glucose is oxidized by glycolysis. Adipose tissue will store excess glucose as fatty acids through fatty acid synthesis. It will also use TAG synthesis for extra storage. Dietary fats are transported by chylomicrons and stored in the adipose. Overall, glucose is obviously a very vital part of metabolism. Glucose is needed for many pathways, including glycolysis, the citric acid cycle, fatty acid synthesis, glyconeogenesis, gluconeogenesis, and many others. Glucose is also needed to produce other vital compounds, including but definitely not limited to acetyl-CoA, ATP, NADPH, NAD+, and FADH. In tumors, the uptake of glucose is done at a much faster rate than in normal tissues. Because of this, it would further increase the rate of metabolic pathways listed above, such as fatty acid synthesis and the citric acid cycle. Because of this, cell production is increased, allowing for tumors to grow. Cancer cells, surprisingly, use aerobic glycolysis. In aerobic glycolysis, in the presence of oxygen, glucose is converted to lactose. Tumors use the lactate produced in aerobic conditions as an advantage over normal metabolic tissues. Since lactate is increased, pyruvate is also increased by the pyruvate dehydrogenase complex. This is very different from other normal tissues that utilize cellular respiration, which produces 32 to 36 ATP. In cancer cells, the uptake of glucose is done primarily through the glucose transporter GLUT1 in an insulin-independent manner. In aerobic glycolysis, less oxygen is taken up, which results in low ATP. This is advantageous because the ability for cancer cells to uptake glucose in a low oxygen environment and then rapidly divide sets these cells apart from normal tissues. Because of low oxygen and conjunction with low ATP, there is less energy within the pathway. When I first discovered that cancer cells went through aerobic glycolysis, I was perplexed. But then I realized that the reason why cancer cells grow and metastasize so quickly is because the cancer cells make up for their low energy producing pathway by going through glycolysis faster than in normal cells. This means that cancer cells will uptake glucose much faster than in normal tissue. In the image provided, you can see that the large glioblastoma tumor uptook more glucose at a much faster rate than the normal tissues. This is evident by the dark red circle, which represents an accumulation of glucose in the area where the cancerous tumor is present. For the next case study, I will be responding to case study number 4. In this case study, a 2-month-old is presenting with jittery spells several hours after breastfeeding, hypoglycemia, and an enlarged liver. So, when overlooking the patient's symptoms and lab results, it seems as if they were in line with glycogen storage disease. So, a glycogen storage disease is a genetically inherited disease and is the result of increased glycogen in the tissues. In a glycogen storage disease, glycogen buildup is caused by glycogen not being properly stored and released. Since the glycogen cannot be released, this results in a decrease in glucose levels, in blood glucose levels, which is hypoglycemia. The accumulation of glycogen in the liver could cause enlargement, as seen in this case. The reason why epinephrine did not impact the blood glucose, but did impact the blood lactate level, is because the glycogen in the liver was not released, therefore causing an accumulation of glucose 6-phosphate. The glucose 6-phosphate enters glycolysis, which increases the lactate levels. Since no glycogen was released into the bloodstream, blood glucose levels did not elevate. Blood glucose levels did increase after feeding because breast milk provides glucose and galactose, which assists in providing extra nutrients for the 2-month infant. When the 2-month-old is in the fasting states, the glycogen is being stored instead of released from the liver, causing hypoglycemia and jittery spells.